Bismillahirrahmanirrahim. O-level biology paper 4-2, and this is the June 2024 exam, which is the most recent exam. The time, as you all know, is 1 hour for every paper 4, or which used to be called the paper 6. It is 1 hour. Now let's start with question number 1. A student investigated the structure and nutrient content of a flower. Figure 1.1 shows the flower. Some petals have been removed so that the internal structure can be clearly seen. Make a large drawing of the flower as it appears in figure 1.1 in the space below. Now, I've tried to do it, might not be a very good drawing, I'm sorry. Now, there are 5 marks for this drawing. What are the 5 marks for? Number 1, clear and clean, continuous lines drawn with a sharp pencil, no shading anywhere. So please do not do any shading or something like this to show something. It has to be very clear. It should be at least more than 100 millimeter wide and correct orientation. Then 3 stamens drawn. Now you can see here I've drawn the 3 stamens, 1, 2 and 3 stamens. At least one longer than the stigma. So this one is the one which is longer than the stigma. And filaments with double lines and anthers delimited. Style drawn with double line and stigma delimited. 3 separate petals drawn. 3 separate petals drawn. Now I haven't drawn that. I will lose a mark for that. This should have come here. So 3 separate petals drawn, and all anthers and stigma protruding above the petals. All anthers and stigma protruding above the. So this was 5 marks, and I would have probably got 4 marks for that. Right? So everybody needs to be very clear on this, how to draw these drawings and how to just practice, and you'll, I'm sure, improve every day. Then it says on your drawing, draw a line and label it P to show where the pollen must land when the pollination takes place. It said using a label line. So you use a label line to show where it will land on the stigma. So what is pollination? Is the transfer of pollen from the anther to the stigma. So label line on top of the stigma would get you a mark for that. Then going back to it, says that the student tested 2 parts of the flower A and B for their nutrient content. The students test both parts with Benedict solution, biuret reagent and iodine solution. Draw a table image to record the results of the students' tests in the space below. So now we can make a table for this, and we have to show that student tested with Benedict's, biuret and iodine solution. And it's saying in the question. Now when we make a table in 3 columns. We have to just test A and B. And then we have to test with Benedict. So what will be the color and what will be the result? If the Benedict solution is blue, that means no reducing sugar present. If the biuret student is blue, then no protein present. If the iodine is brown, then no starch present. So you have to give me the color and then the result. So the student tested, draw table finished to record the results of the student's test in the space below. So this was for 4 marks. Then it says at the end of the test, the students noted that A tested positive with Benedict's and negative with biuret and iodine solution. So B tested positive with biuret and iodine solution and negative with Benedict solution. In the table you have drawn, record the colors of the students would have observed at the end of each test. Now, of course, they're expecting you to now fill in this. So what will be the color of A and what was wrong with A? It says part A tested positive with Benedict's. So what would be the color? It would be red. And negative with biuret. So this would be blue. And iodine. So this would be brown. Right? And what happened with B? B tested positive with biuret. So this would be purple. Positive with biuret and iodine. So it would be black. And negative with Benedict's. So this would be blue. So then this is checking whether you knew what your colors were when you do the food tests. What is the original color of the reagent, and then what does it become when it tests positive or negative? It remains. Benedict's is blue, will remain blue if it's negative. Biuret is blue, will remain blue if it's negative. Iodine solution is brown in color, it will remain brown. if no starch is present. Benedict solution tells you about reducing sugars. Biuret tells you about protein. Iodine tells you about starch. It says state the nutrients present in A. So A was positive with reducing sugar. So it contains reducing sugar, you could have said. It contains glucose, you could have said. It contains maltose, you could have said. Or you could have said it contains reducing sugar. So reducing sugars are glucose is a reducing sugar. Then part B, nutrients present in part B. You can see there are 3 marks for this question. So they are actually somewhere you got to give 2 points. So it was blue, purple, and black. So B contains what? It contains protein and it contains starch. That's why there were 3 marks. There's 1 mark for this and 2 marks for this. So B contains protein, 1 mark. B contains starch, another mark. A contains reducing sugar, or if you had said glucose, you got a mark for that. Then it says C part, figure 1.2 is a photomicrograph of a pollen grain. Measure and record the diameter. You see it is magnified 400 times. So this was about 25 millimeter. They've given you this unit. Then you said calculate the diameter of the actual pollen grain. So what did you have to do? You have to divide 25 by 400 and you give the correct answer to 2 decimal places. So 25 divided by 400 and then you give me the correct answer to 2 decimal places. Now you divided by 400 and then you said correct answer to 2 decimal point 06 and then you gave me the units because here it was 25 millimeter. But you could have multiplied by 1000 and then you'd have given me the answer in micrometer. So any units which you are okay with it, you could have given it to me in millimeters or, but the third mark is for the units. So if you didn't write the units, well, that's a very sad thing if you didn't write the units because that shows how very, very pathetic and careless you are. Then it says figure 1.3 is a photomicrograph of pollen grain from a different species of plant. Describe 2 visible differences in structure of the pollen grain in figure 1.2 and 1.3. It says visible differences. Figure 1.3 and figure 1.2. So you give me the visible differences between the two. So look at it again and look at it again and then say visible difference. That's something very, very common that they give you this sort of a question. So the visible differences were round, oval, the shape, spikes absent, spikes present, and internal division and no internal division. Now what does this mean? You can see this is the internal division. It's sort of divided into 2 lobules or something like that. So there was no such thing here, and so there's internal division and no internal division. Now coming to question number 2, a student investigated the number of plants on a school field. This was done by examining small samples of the field using a square frame. Now that's called a quadrat frame, and measuring 0.5 into 0.5, that's 0.25 meter. So figure 2.1 shows the plants in one of these 0.5 meter into 0.5 meter samples. So we have 3 species A, C, D, and E, and there are a number of these. And species E, I have counted them, and I've written the numbers on it. Then it says the sample contained 3 different plant species. Fine, we look at it and we think about it. The student counted the number of plants of each of these species in this sample. One plant was not fully in the square. You can see this one is not fully in the square, within the square frame. Suggest and explain what you would do about this plant. Well, 2 things. One is that you can count it because more than half is inside the square. Or you can say, no, we cannot count it because we cannot count half an individual, so we will exclude it. It is not fully in the square. And we cannot count a plant because it's not fully inside the square. So either you gave me whatever reasons. I would have, of course, counted it because it is more than half inside the square. More than half of it is inside the square, so I would count it. But you could have said either of these, as I will not count it because it's not a complete plant. So it's either whatever you felt was correct, you would write that. More than half in the square, so include it. Now we come to the next part. Count the number of plants of species E in figure 2.1, taking into account your answer to A. Record your answer in table 2.1. So there are 10 of them. Now you can see how the number of plants C were 7 and then they're 5,600. So basically they've multiplied it by 800. So this has been, you could have even figured this out from here. Then it says the whole field measured 10 meter into 20 meters using the sample to estimate the number of plant species in E in the whole field. So it is 10, so 10 into 800. And then it says record the value in table 2.1. So please record it here, and it is 8,000. So recording it, so if there are 10 in each sample, so there'll be 8,000 plants. And but how did you do a reference to 800 samples? You could have said here 800 samples, because there are 2 marks for this. So 800 samples, 10 in each sample, so there will be 8,000, and then you recorded that in the table. So that got you your 2 marks. Explain why the student counted the number of plants in samples of the field instead of counting the number of plants in the whole field. Is too time consuming to count them all, and it's too difficult to keep an accurate tally. More likely you're going to make a mistake. So even if you did, this is a very good representative figures. You see, you do these quadrats and you place them on the field, and then of course you add up all of them, and then you make a general estimate of the number of plants on that entire field. So too time consuming to count them all, or you could have said too difficult to keep an accurate tally, or you could have said more likely to make a mistake. Then it says suggest 2 reasons why taking several samples would improve the accuracy of the estimate for the number of plants in the whole field. If you only took 2 samples, well, that would not be very representative. Maybe it's a part of the field where there's some shade or there's some tree nearby, and so that's not very representative of it. So suggest 2 reasons. The plants are not evenly distributed. Larger proportion of the field is sampled. More representative, you are more likely to include every type or every number of plant species. So plants not evenly distributed. Suggest 2 reasons why taking several samples would improve the accuracy of the estimate for the number of plants in the whole field. So several samples, how would they improve the accuracy? Is plants are not evenly distributed. Larger proportion of field samples would be more representative of so larger samples means you are more likely to include every type of plant species. Now, some of you might know it again, but you don't know how to word it, so you need to improve your English. We're going to take the exam in English, not in Urdu or Punjabi or Pashto. Then coming to the F part of the question, use the data in table 2.1 to construct a bar chart. Please remember, a bar chart is not connected. Bar chart should not be connected, and there should be spaces. A histogram is one in which it is connected. So bar chart, you have to do bar chart for C, D, and E. I'm just showing you, I haven't actually done it exactly. So there's nothing on the x-axis, but then you have to decide what is the scale that you take on the y-axis and what would you label on the x-axis and the y-axis. So you all have to be very clear on that. What are you going to label on the x-axis and the y-axis? Bar chart means there's going to be space. There's going to be space between the bars, and the space between the bars should always be the same. In a histogram, you then connect it. So this would be a histogram. This would be a histogram. But we're not doing a histogram, we're doing a bar chart. So I'm just going to now again revisit this. Now the first point is axis fully labeled. So you must label the axis plant species C, D, E, estimated number of plants in the whole field. Now I'm writing this just to get me because it's on the other page. So C was 5,600, D was 3,200, and then E was 8,000. So we've got to get it something to 8,000. So let's look at the scale now. What scale should we use? So the nearest scale that I can think of is 0, and every 2 centimeter is 2,000, 4,000, 6,000. So this would be 4, no, this would be 6,000, yes. And this would be 8,000. So 2, 4, 6, and 8,000. I mean, this is the nearest that I can think of. Maybe you could have done something else. 45, 6. No, you couldn't have done, I think. So, but don't come up with weird things like 1600 or 1400 or something like this. Make it clear it's 2000, 4000, and 8000. Now, C was 5600. So, let's start plotting it now. So, then plotting the bar charts, 5600, you can shade them if you want to. Just put some lines on it. Why I tell you to do it with a pencil? Because if you made a mistake, then you can erase it and do a proper one. It's not a question that we don't accept it if you've done it with a pen. We'll accept it even with a pen, but then you might have drawn it wrongly, you might have taken the scale wrongly, you might have made a mistake. Like just now I did 2000, 4000, 5000, I made this mistake. So 2, 4, 6, and 8, you have to have a pattern. Every 2 centimeters is 2000. So you can't be just doing it. I don't know, some of you make these mistakes when I check your papers. So this would get you your 4 out of 4. Then it says question 3, plants can be provided with fertilizer to help them grow. Some students were provided with germinated seeds of the same species in some shallow dishes and 10% fertilizer solution. One dish is shown in figure 3.1. So they were germinated seeds of the same species in a shallow dish and 10% fertilizer solution. One dish is shown in figure 3.1. The student had access to any other common laboratory apparatus, germinated seed, shallow dish, and we have these. How many of these we have? How many seeds do we have? Well, we haven't counted. Then we're provided with some germinated seeds. Plan and investigation the students could carry out to find the effect of different fertilizer concentrations on the plant growth. So anything which we are going to change, different fertilizer concentration. Now that is what we have decided to change. We could have said give it different light intensity. We could have said give it different carbon dioxide concentration. We could have said give it different nitrate concentration. So whatever we change, that is the independent variable. So what are we investigating? We are investigating plants can be provided with fertilizer to help them grow. So we gave them the seeds and we're saying access to common laboratory to find the effect of different. Does it affect it having a 1% solution, so you say you have a 10%. So now you can't make 11% out of it. So you would make an 8, 6, 4, 2%. You would make 5 different concentrations and you would add the same volume of it to the seeds. And then you would leave them for about you'd leave it for about 6, 7 days. You can't leave it for a short while, for hours. So, and then you would measure the height and then you'd compare the height. So you would say measuring, how would you measure growth? You could have said height, you could have said mass or height at the start and the end. Then at least 3 concentrations, and then they would all be less than 10%. You can't make it more than 10%. You've been given a 10%, so it has to be 8, 6, 4, 2. Same volume of the fertilizer solution, leave for the same amount of time, 6 days, 5 days. Same light intensity and same temperature and same carbon dioxide and same volume of the water added every day. Then repeat them at same fertilizer concentration and calculate the mean. So for every concentration, in 10% you would repeat the experiment 3 times, in 8% you would repeat it 3 times, and in 6% you would repeat it 3 times or 5 times. As I always say, it's better to say 5 times because they say a minimum of 3 times. And then you would compare the data to see does it affect the height? Has it affected the height of the seeds when you have increased the fertilizer concentration? They're not asking you to say yes or no. They're just going to say, okay, then you compare the data. So then we will take the mean height and we will say, okay, at 10% the mean height was this, at 8% the mean height was this. So yes, it does affect it, but to a certain concentration, and after that there seems to be no effect. Or yes, as you keep on increasing the fertilizer, it will increase the height of the plant. So it is a direct linear correlation. Now in the end they've asked you identify the independent variable. And I've always said independent is I, which I have changed. That is the fertilizer concentration. You are changing the fertilizer concentration. That is the independent variable. If you are still stuck with independent and dependent variable, please Google it. There are lots of YouTube videos on that. First of all, you have to realize is if you're going to measure, if you're going to change the fertilizer concentration, what are you going to measure? You're going to measure the growth. So how are you going to measure the growth? You're going to measure either by height or mass at the start and the end. So you've left it for 5 days and you've applied this fertilizer concentration. Every day you water the plant. Yes, you must water it every day because you're leaving it for 5 days. So in one of them, you've added 10% fertilizer and you've added 5 ml of that. Please understand, volume is measured in ml or cm cube, and mass is measured in gram or milligram. Length is measured in millimeter or centimeter. So at least 5 concentrations of fertilizer, and then because you've been given a 10%, so you have to dilute it. You can't say, okay, give me a 20%. No, that's wrong. What is given in the question is a 10%. So you have to dilute it further. So 10%. So, and I've given you these values. You could have written anything. You could have said even 10, 9, 8, 7, 6, 5 or something like this. I'm not saying you have to give it to me, but it has to be a pattern. It is like I have done it an interval of 2, 10, 8, 6, 4, 2. You could have said 10, 9, 8, 7, 6. And same volume of the fertilizer solution. Leave for the same time, 5 days or 6 days. You have to leave it for that long. You can't leave it for a few hours. The hours is not going to result in growth. Then same light intensity. Now you've got to come up with things which are going to affect it. Photosynthesis. Enzymes are working. The plant is growing. So same light intensity, same temperature, or you could have said same CO2 levels, or you could have said add the same volume of water every day. If you're adding 5 ml water every day, then add 5 ml water every day to the plant. Then repeat at the same fertilizer concentration for every fertilizer concentration. I mean for 10% repeated 3 times, for 8% repeated 3 times, for 6% repeated 3 times, for 4% repeated 3 times, for 2% repeated 3 times. And then you compare the data for the height and the fertilizer concentration. Independent variable here was the fertilization concentration. Dependent variable was the height that you were measuring. But what were you measuring? You were measuring growth, but that is not the dependent variable. How are you measuring growth? That is the dependent variable. You are measuring growth by measuring the height of the plant at the start and at the end. So that completes this paper. And thank you very much. And please leave your comments if there's anything which is not very clear.